Next, we're going to be looking at organic compounds. Remember, the basic definition of organic compounds are molecules or compounds that include carbon but bound with hydrogen, because exceptions to the just contains carbon for organic compounds is CO2 or CO, or carbon dioxide and carbon monoxide. So they are inorganic, and that's why at the very beginning, if you go back to part 1 for this video series, you'll see that there were some exceptions to saying that inorganic compounds don't contain carbon and organic compounds do. So better definitions for both are inorganic compounds don't contain carbon with hydrogen, whereas organic compounds do. Because we see carbon dioxide and carbon monoxide, or CO2 and CO. Yeah, they contain carbon, so you'd want to put them with organic compounds, but they do not contain carbon with or in combination with hydrogen. There's no covalently bonded hydrogen to those carbons, so they are not organic and are in fact inorganic. So make sure that mentally you make those adjustments to those base definitions that are in your textbook. Some examples of organic compounds include things like carbohydrates, lipids, which you may know as fats, but that's a little bit of a misnomer, so we do really want to call them lipids, proteins, and nucleic acids. So we're going to look at all 4 of those, but you should know those as kind of the big list, the general polysaccharides, or what are sometimes called the biological molecules. The polysaccharides, or like carbohydrates, or biological molecules like proteins and nucleic acids, and to an extent lipids, they are all polymers. So they are chains of similar units. The monomers are what build up or make up the polymers. For example, if you put a bunch of monosaccharides together, you'd build up a carbohydrate. But a carbohydrate is just a polysaccharide. If you had a lot of amino acids and you bound them together, you'd be on your way to building a protein. And if you had a lot of nucleotides, you would put those together and you'd be on your way to building a nucleic acid. Lipids are a little different. They're pretty varied when it comes to being a biological molecule. And so we don't really say they have a true monomer, but we can say that most lipids contain glycerol and fatty acid chains. We'll be looking not only at these polymers, which are essentially our biological molecules, we'll be looking at them and their monomers and how we can both synthesize these large substances, these large organic compounds, via dehydration synthesis, and how they're broken down via hydrolysis reactions. So let's just start with carbohydrates. Carbohydrates are another term for polysaccharides. Carbohydrates are essentially sugars and starches, so they're readily available or useful for energy. They are an organic compound. They do contain carbon in combination with hydrogen, but also some oxygen. And the hydrogen and oxygen are generally in a 2-to-1 ratio. There's different types of carbohydrates. However, what you're probably used to calling a carb or a carbohydrate, things like bread, those are polysaccharides. But if you just had one single sugar, that's a monosaccharide. And it's the smallest unit of a carbohydrate, and that's why it's the monomer of a carbohydrate, because all disaccharides and polysaccharides are linked sugars. So you have one sugar as our smallest unit. A common example given for a monosaccharide would be glucose, but fructose, galactose, deoxyribose, and ribose are all monosaccharides or simple sugars. A disaccharide is what you would get if you linked 2 monosaccharides together. Then a polysaccharide again is what you would get if you linked a bunch or many sugars together. So mono meaning 1, di meaning 2, and poly meaning many are prefixes to become familiar with, if you're not. So let's look at disaccharides. They're double sugars, but we can get an idea of what the monomers look like as well as the disaccharides by looking at the 2 sugars linked together. So the monomers of carbohydrates again are monosaccharides. When you link 2 of those together, you get a disaccharide. We can see down at the bottom that glucose and fructose make sucrose, 2 glucose monosaccharides make maltose, and then galactose and glucose make lactose. So we're linking monosaccharides or single simple sugars together to make disaccharides in these examples. Now monosaccharides can pass through cell membranes to an extent. They generally pass through channels, but double sugars never pass through cell membranes. They're way too large to pass through phospholipid bilayers. Important disaccharides are the ones listed below, the sucrose, maltose, and lactose. And they are going to be formed by dehydration synthesis, meaning that that bond is formed via the removal of water. And the removal of water causes a bond between the two monosaccharides. Polysaccharides are essentially even more sugars. Really, they're just polymers of simple sugars. Again, the simple sugar is another name for the monosaccharide. Important polysaccharides we're going to look at are starch and glycogen. Starch is going to be found in plants. It's the carbohydrate energy storage form used by plants, whereas glycogen is the carbohydrate energy storage form used by animals. We actually store glycogen in our skeletal muscle and our liver cells, and it's not very soluble, so while in certain rare circumstances a monosaccharide may be able to go through a cell membrane, and disaccharides cannot, polysaccharides would never go through a cell membrane or a tissue. They are very large structures and would need to be stored in certain locations. So here's an example of a polysaccharide. And we can just see that there are many monosaccharides linked together via dehydration synthesis. And a single sugar, if you haven't picked up on this yet, forms a ring. So a monosaccharide, which is the monomer of a carbohydrate, which are essentially polymers or polysaccharides, they have ring structures. Next, let's look at lipids. So lipids also contain carbon and hydrogen and even oxygen. They are organic compounds, so the carbon and hydrogen make sense, but the proportion of oxygen in lipids is going to be lower than in carbohydrates, so we won't see that 2-to-1 ratio. Lipids are completely insoluble in water, and all that means is that oil in water, or fats in water, or essentially now lipids in water, don't mix, and you've probably seen that many times. The main types of lipids that we're going to look at are triglycerides, or neutral fats, phospholipids, and steroids. Triglycerides are definitely going to be some of the first substances we look at. But before we get there, we want to just separate kind of what two terms mean. There are saturated fatty acids and unsaturated fatty acids. And both phospholipids and triglycerides contain fatty acids. And if there is just a single bond between the carbon atoms that make up the fatty acid tails of, say, a phospholipid or a fatty acid chain, then we would say that those fatty acids are saturated because they have the maximum number of hydrogens covalently bonded to their carbon atoms. Saturated fatty acids are also solid at room temperature. So saturated, solid, a good example of a solid saturated fatty acid would be butter. Whereas unsaturated fatty acids have kinks in their fatty acid tails. They don't have as many hydrogen atoms as our saturated fatty acids, so they're missing a couple, one or two, because they have one or more double bonds between their carbon atoms, which takes up one of the available bonds or electron pairs that the carbon atom could use to bond with a hydrogen. So that's the reason for the reduced number of hydrogen atoms. Unsaturated fatty acids are also liquid at room temperature. So plant oils like olive oil are an example of unsaturated fatty acids. And again, unsaturated fatty acid chains have kinks in their tails or slight bends, whereas saturated fatty acids are straighter. So words to associate with saturated fatty acid are saturated with solid, as far as what type or consistency the fatty acid chain would produce. Also, saturated fatty acid with straight, so saturated, straight, because the tail's going to be much straighter than that we see with unsaturated fatty acids. So here is the triglyceride formation. What we see here is a bunch of saturated fatty acid tails. None of them have double bonds between their carbon. There's no kinks. We see that in order to build a triglyceride, we had to combine glycerol and fatty acid chains. And that's why in general, we can say that lipids commonly have glycerol and fatty acid. But they don't have a true monomers, because things like steroids aren't going to necessarily have fatty acid chains, or they might look slightly different than things like phospholipids or triglycerides. Phospholipids look a lot like triglycerides, but they don't have these three fatty acid chains, which is where a triglyceride is getting its name from. So it's three fatty acid tails bound to a glycerol molecule, so that's how we're getting triglyceride. Whereas phospholipids have two fatty acid tails. One is unsaturated because we can see the kink, but also the double bond between the two carbons. It still has a glycerol backbone, but it also has a phosphate group. a phosphorus-containing group. Steroids tend to be rings. They tend to have anywhere from 4 to 5 rings, and sometimes they'll have functional groups attached. Common steroids that we're going to be talking about are things like cholesterol, vitamin D, which must be activated in the human body. In AMP2, you'll talk about steroid hormones like estrogen and testosterone, and even bile salts when you get to digestion. But the most important steroid is cholesterol, because all steroid hormones and other derivatives are going to be derived from cholesterol. It's kind of our starting point for steroids. So for steroids, again, we're looking for kind of 4 interlocking rings, but definitely ring structures, oftentimes with functional groups attached. Estrogen, testosterone, vitamin D, cholesterol, those are some terms you should know are listed or commonly recognized steroids. And the reason that cholesterol is the most important steroid is it's the building block not only for vitamin D, but for bile salts, for steroid hormones, and it's important in cell plasma membrane structure. So it's going to influence, essentially, how fluid the phospholipid bilayer is in cells. So it just has a lot of functions. Next, we'll move on to another organic compound, or polymer. Proteins are a biological molecule that comprise about 20 to 30% of cell mass. One common function of proteins is to act as enzymes. Not only are proteins structural, but they're functional in that they catalyze reactions. They can also cause muscles to contract. They contain not only carbon and hydrogen, which make them organic compounds, also oxygen, but even nitrogen. And if you remember back to the beginning of the chapter 2A video lectures, carbon, hydrogen, oxygen, and nitrogen are going to be the substances that make up about 96% of the human body. And then in trace amounts, you'll find some sulfur and some phosphorus. Now, for proteins, the monomer is amino acids. So a polymer of amino acids is saying something built up of only amino acids, and that's going to be a protein. The bonds that hold amino acids together in order to build a protein are peptide bonds. And these peptide bonds would be formed via dehydration synthesis. We're going to be looking really in depth at proteins. We're going to be looking at their shape and their function, and those things are both due to 4 structural levels of protein organization. But let's get those basics down again before we move forward. So amino acids are the monomer of proteins. Amino acids are essentially going to be linked together via peptide bonds. We're going to look at a base amino acid in a minute, kind of a generalization, but we'll see that amino acids have 2 important functional groups, an amine group and a carboxyl group. And these amine and carboxyl groups will be on one or the other side of the amino acid, and then there's always an R group on amino acids. And this R group is what changes in each amino acid. It's why we have 20 different amino acids, because they all have an amine group and they all have a carboxyl group, but their R group varies. And that is another term for the functional group, the R group. Proteins, again, are combinations of amino acids. Again, we have 20 types. I'll show you an image in just a moment. We tend to say that a small protein is really just a polypeptide. That's 10 to 50 amino acids bound together. Whereas our big macromolecules, our biological molecules that we're calling proteins that have large functions or very structural, they contain about 100 to 10,000 amino acids. So here is that basic amino acid. We can see our R group, also known as the functional group, and then the general component of every amino acid is going to be on one side an amine group, which is a nitrogen covalently bonded to 2 hydrogens. Then on the other side, a carboxyl acid group, here just being called an acid group. Carboxyl acid group is a carbon double bonded to oxygen and single bonded to a hydroxide ion. So OH there is going to be an oxygen that's bound to hydrogen covalently. Here are the 20 amino acids. If you ever take an upper level biochemistry class or cell biology or both at the same time, which is common in biology degrees, then you'll probably have to memorize not only all 20 amino acids, but their abbreviations. There's also single letter abbreviations for the amino acids, and oftentimes classes like that would ask you to memorize or even be able to draw their functional groups. We're going to stick to basic structural organization of proteins, and there are 4 levels of protein organization. And this really determines the shape and function of your proteins, whether your protein goes on to be structural in function or whether it goes on to be something like an enzyme. We still would see at least up to the third tertiary level of organization for all proteins. The primary level of organization for proteins is just the order that you link the amino acids together, the linear sequence of amino acids, kind of like beads on a string. If we're going to link amino acid 1 to 3, and then 3 to 2, and then 2 to 4, just what order are you linking the amino acids in? Because there's 20. Secondary structure is going to be how those primary amino acids, so how that order interacts with the rest of the beads on a string. So basically how the entire string of amino acids acts towards each other. Because sometimes the primary sequence likes to form a helix. It likes to coil around itself. Other times it forms kind of a wavy pleated sheet. Depending on its secondary structure, it'll look a little bit different. Then, if you happen to have part of your secondary structure as a helix and part of your secondary structure as a pleated sheet, and then that folded in on itself so that the helix and the pleated sheet were right next to each other, it kind of started to basically become globular, then you'd be looking at tertiary structure. Because tertiary structure is how the secondary structures interact or orient around each other. And then quaternary structure. Now, not all proteins make it to quaternary structure, but really large proteins do. Proteins like hemoglobin, for example. Quaternary structure is when you have 2 or 3 or more tertiary proteins that combine together. It's how those different polypeptides, those different tertiary structured proteins interact with each other. Hemoglobin was the example I gave because it actually has 4 tertiary structured proteins combined together. You have 2 alpha and 2 beta chains of hemoglobin, and all 4 of them are their own tertiary protein structure. So let's look at these with pictures. It'll make a little more sense. So I oftentimes call the primary structure beads on a string because you're just linking the amino acids together in a certain order. So here's an amino acid, here's an amino acid, here's another. So it's pretty boring, but it's pretty basic as well. So it's just the sequence of amino acids that have formed the polypeptide chain, the protein chain. Now, I know they're showing you the molecular formula, but still picture our beads on a string. So we had 1 bead, 2 beads, 3 beads, 4, and so on. They're still there. They're just showing you the molecular structure. So imagine your beads on a string started to coil, or maybe your beads on a string started to zigzag, form a pleated sheet. That would be secondary structure, essentially when your primary structure starts to take on shape. Tertiary structure is going to be when your secondary structure starts to interact. So here we have an alpha helix that is right next to a zigzaggy pleated sheet. You start to see that in tertiary organization that the protein starts to look really globular. Not large and necessarily very globular, and I'll show you what I mean by very globular in just a minute, but it just starts to look kind of clumpy. It's no longer beads on a string, it's no longer just slightly coiled or zigzagged. It starts to kind of take on another shape, a shape that's kind of more full-bodied. So essentially superimposed on secondary structure when the alpha helices or the beta sheets are folded up in kind of a compact globular molecule, that is tertiary structure. Here's quaternary structure. So you can see we have on the left 1 tertiary structured protein, another tertiary structured protein, and they've come together, they're interacting right here in the center, and they've formed a quaternary structure. So again, when 2 or more polypeptide chains that are in their own tertiary structure combine or interact to form a functional protein, then that is a quaternary structure. It does not always happen. Some proteins only ever exist or make it to the tertiary structure. Here you can also see a space. If this, for example, was a protein that was an enzyme, this space would probably be for something to bind. A lot of enzymes take on a particular shape in order to match what they're trying to bind to or what they're trying to facilitate in the form of enzyme reaction catalyzing. So just a summary of what we've been looking at. We know proteins are made of amino acids because amino acids are the monomer of proteins, which are going to be polymers. They have structural and functional properties. We've talked about enzymes. An example of a really large quaternary protein was hemoglobin. We know that proteins are found in muscles. What you find in muscles are actually myofilaments called actin and myosin, and they allow for muscle contraction. And a lot of the structural material of the cell is proteins. So they have by far kind of the largest function in cells. And then we just looked at the structural levels of proteins, which essentially are fancily named 1, 2, 3, and 4. But instead of that, we would say primary, secondary, tertiary, or quaternary organization. So let's continue on and just talk more about those functions for proteins. The shape of a protein, depending on its organization, could either be fibrous or globular. And it's exactly what you think. Fibrous proteins tend to be structural, globular proteins tend to be functional, and fibrous structures tend to be strand-like, whereas globular proteins tend to be compact, spherical, globular, for lack of a better phrase. So while fibrous proteins are strand-like, water-insoluble, and stable, most of them are going to make it all the way to quaternary structure. They're at least going to have tertiary structure, but they just provide mechanical support and tensile strength. There are things like elastin and collagen and keratin, which are some of the things you find in epithelial cells. They just give structure and body and shape to cells. Certain contractile fibers like actin and myosin would also be included under that fibrous protein category. Now, globular proteins, we say they're functional because these are the proteins that become enzymes. They're also tertiary quaternary structured. They also have specific functional regions. That area I said where this is a quaternary protein a couple slides back and it has a space, and this is probably where something would bind if it was an enzyme. Those are called active sites. Those specific functional regions where something is expected or would bind is an active site. What would bind to an enzyme at this active site is called a substrate. But there are other things besides enzymes that are functional, things like antibodies, which are immune proteins, hemoglobin, which carries oxygen, hormones, which have many functions in the human body, and even molecular chaperones for things that are insoluble in the watery environment of things like blood plasma or other tissues. But enzymes is still going to be our major focus for our functional proteins. So here's just the two basics. Our fibrous on the left, very strand-like, long, supportive, structural in nature. Globular protein on the right is actually hemoglobin. And hemoglobin, again, is a quaternary structure. We can actually see the 4 polypeptides that are in tertiary structure making up this quaternary structured globular protein. Here's a quaternary protein that is very involved, very complex, definitely globular, but we can also see that there are centralized areas in its periphery and also in its middle that perhaps are active sites ready to bind to a substrate. All proteins can also denature. So essentially what this means is all proteins can unwind. And when they unwind, they lose their function because usually the shape, that organization of the protein is important for its function. So shape change and disruption of active sites, or even just shape change of fibrous proteins, changes like increases in temperature or decreases in pH, or vice versa. All of these things can denature or unwind a protein. And especially with the globular proteins, if you unfold them, they lose their functional 3D shape, and therefore they lose their ability to work. A lot of times denaturation is reversible, especially if it's only temporary. If the temperature just increased quickly but came right back down, then denaturation is reversible. And if normal conditions, normal pH, normal temperature are restored, then the protein will be restored. But if there are extreme changes in temperature, pH, other external environments that are generally around an enzyme, for example, then you can't reverse denaturation. A common example is the cooking of an egg. If you've ever cooked an egg, you know that you can't uncook the egg. You also know the egg starts out as a little bit more liquid than it does when you are done cooking it, and it's much more solid. That's a denaturation process where you have changed the proteins in an egg by applying heat, and by doing so, you have changed their structure, and they no longer have the same function they did when they were in the liquid state now that they're in the solid state, and that is not reversible. Enzymes are probably the proteins we're most worried about becoming denatured because enzymes, while globular proteins, are definitely functional, but they're also biological catalysts. They're important for reactions in the human body to take place. Anything that's a biological catalyst regulates and essentially increases the speed of chemical reactions. So without biological catalysts, without enzymes, a lot of reactions in the human body wouldn't take place fast enough for you to continue on living. Catalysts are interesting because they don't get used up in the process of making sure that reactions proceed as they should. And not just proceed as they should, but proceed quickly. What enzymes do are lower the energy needed to initiate a chemical reaction. That energy is called activation energy. And because they lower the energy needed for a chemical reaction to take place, that means the chemical reaction can take place quicker because less energy was needed. And again, this is what allows for millions of reactions per minute in the human body and essentially allows or sustains life. So just a quick mini lesson on enzymes. Enzymes are an example of a functional globular protein. Another name for enzymes are biological catalysts because they catalyze chemical reactions. They make chemical reactions go faster because they lower the activation energy, which is just the amount of energy needed for a chemical reaction to occur. Enzymes are also specific. They don't just bind to anything. They bind to specific things. What they bind to is a substrate. Substrates bind to an enzyme's active site, and enzymes are completely unchanged once they are done catalyzing a reaction. Oftentimes, enzymes have names that end in ase. For example, hydrolase, oxidase, lipase, amylase, catalase. Those are all examples of enzyme names. So if you ever see a word that ends in ase, you have no idea what it is, and you're having to guess, enzyme is a good guess for what that substance is. There's 3 general steps involved in an enzyme action, and that is its substrate, what it's specific to, binds to its active site. That forms an enzyme substrate complex, which is just the enzyme bound to its substrate, them together. And then that complex is going to undergo its chemical reaction. So essentially the enzyme is going to hold the reactants close together so they can either be broken down or put together. So we can either break bonds or build bonds, and then whatever we make, our product is going to be released from the enzyme, released from the enzyme's active site. So here we have on the left a chemical reaction trying to proceed forward or occur without an enzyme, and we have a large activation energy that must be overcome in order to get our product, to go from reactants to products. You can think of this as we need a lot of energy to get our reactants over the hill without an enzyme, whereas with an enzyme, the hill is lowered, so we don't need as much energy to get our reactants over the hill in order to form product. But with the enzyme, what's happened actually is we've lowered the activation energy that's required in order to allow the reaction to proceed forward or occur. Here's a picture of an enzyme that's working. So we have in purple our enzyme. In orange, we have our substrates. This is just what the these are amino acids, but this is just what the enzyme is going to bind to. They are going to bind at the active sites. So substrates bind to the active sites of enzymes. So when the enzyme and substrates come together, we form the enzyme substrate complex, or the ES complex. Energy will be used to either make or break a bond. In this example, we are making or building a bond. So this is an example of dehydration synthesis, where water is removed in order to make or build a bond. So this is also an anabolic reaction. Once we have performed our chemical reaction, the product, because now it's not the same as the substrates, it's not the reactants anymore, it's a new product, it will be released from the enzyme. And the enzyme is fine. It's not destroyed. It's perfectly okay to go on and do this again and again and again. So enzymes speed up or catalyze chemical reactions by allowing substrates, which are reactants, to bind to active sites and basically exist close to each other so that they're more likely to perform or go through a chemical reaction in a faster manner. And then our last organic compound is nucleic acids. And nucleic acids are organic compounds, so we should expect to see carbon and hydrogen, and especially oxygen now, and nitrogen's also there, and then in trace amounts phosphorus. The monomer of nucleic acids is nucleotides, And what nucleotides are made up of is a nitrogen base, a pentose sugar, and a phosphate group. And that's an easy question to put on a test. A question could just be, which of the following make up nucleotides? And again, you're looking for these three things: a nitrogen-containing base, a pentose sugar, and a phosphate group. Sometimes students will abbreviate that and just look for base, sugar, phosphate. And that is helpful if you want to kind of abbreviate what you're looking for. There are two major classes of nucleic acids, and you're probably familiar with at least DNA, or deoxyribonucleic acid. And there's also RNA, which is ribonucleic acid. We have both in our cells. Ribonucleic acid is actually a great transport molecule, but also able to work in some other capacities in order to allow us to read DNA and make protein. There are going to be 5 nitrogen bases contributing to the nucleotide structure. We're going to look at them in just a minute. I've listed out all of their abbreviations. So for adenine, you would use the term A. For guanine, which is spelled Guanine, you would use G. For C, it's cytosine. For T, it's thymine. And then the U stands for uracil, which is URACIL. So I'll just read the whole name again: adenine, guanine, cytosine, thymine, and uracil. And uracil is only found in RNA. Instead, in DNA, you would have thymine. So you don't find thymine in RNA. You find uracil instead. Let's look at DNA and RNA separately. So DNA is a double helix. You find DNA in the nucleus of the cell. It's always in the nucleus. It should not be out of the nucleus, especially in human cells, which are eukaryotic cells and have a true nucleus that contains that genetic material. DNA is definitely that genetic blueprint. We're going to use it for things like making proteins. And DNA nucleotides contain either guanine, cytosine, thymine, or adenine, because remember, uracil is only found in RNA. Also, DNA nucleotides contain deoxyribose as their sugar, and they still contain phosphate. DNA is also going to exhibit complementary base pairing. So for example, since we have 2 strands of DNA and they're both made up of linked nucleotides, at one point the bases come together and they will hydrogen bond in order to form what is called the double helix, which is essentially 2 strands that have twisted around each other. In the double helix, the adenine base of one strand always pairs with the thymine base of another strand. And then cytosine of one strand always pairs with the guanine of the other strand, and vice versa. So A always pairs with T, and G always pairs with C, because the bonding of the nitrogen bases, which will form the middle or kind of the rungs of the DNA ladder, are very specific in how they pair together. So here we have our bases in the center of DNA. Think of it as a twisted ladder. The bases are going to be the rungs, and notice that C always pairs with G, and A always pairs with T. So come up here, you'll see that a little bit better. A always pairs with T, C always pairs with G. And these dots right here stand for hydrogen bonds. And even though hydrogen bonds are weak bonds, even though hydrogen bonds are between molecules, so they're intermolecular, if you have a lot of them, then you have a strong bond. Hydrogen bonds in abundance are very strong. By themselves, not so much. And then if you're looking at these purple parts of DNA, wondering what they are, the purple parts of DNA are representing the backbones of DNA, and the backbones of DNA are made up of the other 2 parts of a nucleotide. So we already looked at the bases, so we would look for sugar and phosphate. So oftentimes these backbones, or the sides of the DNA ladder, are called the sugar-phosphate backbone. So for DNA, it's a double helix. It's found in the nucleus. It contains the bases A, T, C, and G. The rungs are going to be the bases, and the sides of our ladder are going to be sugar and phosphate groups. And we know that makes sense because DNA is made up of nucleotides, and nucleotides are made up of a nitrogenous base, a phosphate group, and a sugar. It's a pentose sugar, but that just means it has 5 points. And we can actually look at a sugar up here. 1, 2, 3, 4, 5. So now let's look at RNA. RNA is ribonucleic acid, and whereas DNA had deoxyribose as its sugar, RNA has ribose as its sugar. Both deoxyribose and ribose are monosaccharides. RNA is also a single helix, so it's a single-stranded molecule. You can find it in the nucleus, but you can also find it in the cytoplasm of a cell. So it's not sequestered away or kept in the nucleus like DNA is. It can be in either location, but it's inside the cell. Instead of having thymine, you would have uracil as a base. So you'd have guanine, cytosine, uracil, and adenine as the bases that you find in the nucleic acids of RNA, or the nucleotides of RNA. There's also 3 different types of RNA, and in Chapter 3 we'll look at them in a lot of detail, but the 3 types of RNA are messenger RNA, abbreviated mRNA, transfer RNA, abbreviated tRNA, and ribosomal RNA, abbreviated rRNA. And they have different functions for the cell, and again we'll talk about them in more detail. A single nucleotide that is very useful in the human body is adenosine triphosphate, or ATP. This is the energy molecule for cells. It's a source of immediately usable energy for a cell. Again, that's ATP, adenosine triphosphate. Adenosine triphosphate is essentially just chemical energy. You get this chemical energy when glucose is broken down or oxidized, and the energy released from its oxidization is going to be captured. ATP again stands for adenosine triphosphate, and it powers pretty much most chemical reactions in cells, any that are not passively powered. The structure of ATP is similar to nucleotides. It contains a nitrogen-containing base, in this case adenine. It contains a phosphate group, and it contains a sugar. Now it's similar to an RNA nucleotide, so its sugar is ribose. Here's the structure of ATP. You have adenine, that is the nitrogen-containing base. Look at all the nitrogen. You have ribose, which is a 5-point sugar. And you have 3 phosphate groups. So just one would qualify it as a nucleotide, but we have 2 additional phosphate groups in ATP. So adenosine triphosphate, the tri is for 3 phosphates. If you were to break this phosphate off, the breaking of bonds is exergonic, and that would release energy. So that would release energy to power a chemical reaction. And ATP is useful because you can do it again. Even after it's lost the 3rd phosphate, you can break off the 2nd and release even more energy, because these last 2 terminal phosphates can both be hydrolyzed to release energy. If you break off 1 phosphate, you're left with 2, so it's adenosine diphosphate. Then if you're going to break off both, you're left with only 1 on ATP, which is no longer ATP, it's adenosine monophosphate. So AMP, ADP, and ATP are going to represent hydrolyzation stages of ATP. Here is an example of its breakdown and building. So its breakdown is going to be hydrolysis. So here's ATP. So it's going to be the nucleotide with 3 phosphates. You add water to the reaction, it'll dissolve or break the bond between that last terminal phosphate, and you'll be left with adenosine diphosphate plus a singular phosphate all by itself. When the phosphate is like this with an I, which is written as a subscript after the letter P, this is called inorganic phosphate. It's a free phosphate, and it's going to suggest that energy has been released. And that's what you're seeing here to finish or complete the reaction. We could go back in the opposite direction and make ATP, and this would require energy to put the inorganic phosphate back on ADP. So this is going to be an endergonic reaction that absorbs energy and proceeds to make ATP through dehydration synthesis, or the removal of water in order to make a bond. Some common functions or uses for ATP include transport work. In Chapter 3, we're going to look at active and passive transport. Active transport is going to use ATP in order to move things against their concentration gradient through channels. We'll also look at mechanical work eventually, especially when we get to chapter 9, and we'll see how ATP powers the contraction of not only smooth muscle, but also skeletal muscle and cardiac muscle. And there's plenty of chemical work that will be suggested is powered by ATP throughout this entire course. We'll not always take something all the way down to its molecular basics, because there's so much to discuss in anatomy and physiology, but be sure that ATP does phosphorylate or get added onto, or dephosphorylate, get taken off of, many different ions and chemicals in order to facilitate a chemical reaction. And that's it for Chapter 2, Part B.